This is Biology 1, Chapter 12, Part 5 on Molecular Genetics. In this portion of the video lecture, we will be going over the genetic code and the process of translation, which is where we take the information from RNA and make proteins from that sequence. When we talk about the genetic code, and we talk about what these genes code for. Codons are the 3 base code in DNA or messenger RNA. So a codon is 3 bases. It would be, for example, adenine, adenine, guanine. That would be one particular codon that would code for a particular amino acid. Remember, amino acids are the building blocks of proteins. So these codons code for the building blocks to make the proteins. Now there's 2 different ways that we can decode what is read. The picture there on the left side, that's what your book uses. I don't like this chart. It's very confusing, and basically you end up scrolling through the whole thing to look for the particular codon that you need. On the right side is what I like to call the wheel O amino acids. And you will have a copy of this to use for your DNA decoding practice. And you will also have a copy of this on your test, because I don't really think that it's necessary to make you memorize the 64 codons for the 23 different amino acids. But the wheel is used very simply. The middle part of the wheel is the first letter of the codon. The second wheel is the second letter. And the third wheel will lead you to the name of the amino acid that codon is coding for. So for example, the amino acid GCA is the amino acid alanine. Now you'll also notice that GCU, GCG, and GCC are also codes for alanine. Again, your body has a lot of ways to make sure that if a mistake is made, it's not going to make a difference in the overall functioning of the organism. The genetic code, believe it or not, is called a degenerate code because there's actually more than one codon that will code for a particular amino acid. That way, again, if there's a goof up in transcription or replication, more than likely the mistake is usually made on the 3rd base of a codon. So this way, if there is a mistake made, it's not going to make any difference to what the ending result for the amino acid will be. So if there is an alanine in the amino acid sequence, it can be coded for in a few different ways. So translation is the process of putting those proteins together. When you translate something, you're moving to a different language. And the language that's being transferred is the language of amino acids to the language of whole proteins. And the tRNA, the transfer RNA molecule, acts like an interpreter of the messenger RNA sequence. Remember, transfer RNA has 23 different types, and each one of those 23 different types is linked to a specific amino acid. Putting amino acids together is making a protein. So replication is making more DNA. Transcription is how we make RNA from DNA. And then translation is how we take that message from the RNA and make it into proteins. So the first step in translation, again, the messenger RNA that has the message needs to deliver the message. So it's going to move out of the nucleus and go to the ribosome. Now, transfer RNA works with what's called anticodons. Anticodons are not evil or dark in any way. They're simply the opposite, or they are complementary to codons that are found on messenger RNA. So messenger RNA has the 3 base codon, whereas transfer RNA has a 3 base anticodon. So if the messenger RNA reads of the sequence GAC, that's the codon for aspartic acid. What the transfer RNA will read is CUG, and then it will grab the correct codon that's needed for the sequence. So after we have the messenger RNA moving out of the nucleus and going to the ribosome will attach to a start code. There's a particular amino acid that's used as a start code. It is the amino acid methionine, which is the 3 bases of A, U, and G. So once the ribosome reads AUG, that's where it knows when to start. It'll start bonding those amino acids together. So on the ribosome, the first anticodon from transfer RNA is placed in what's called the P site, or the peptidyl site. The A site, or the aminoacyl site, is where the second anticodon is placed by the transfer RNA. Basically, then what happens is those anticodons join together. So those amino acids will join together by a peptide bond into an amino acid chain, which is a protein. So this process will continue until a certain stop code is reached. So when the ribosome reaches a stop code, and there are 3 stop codes, UAA, UAG, and UGA. Once the ribosome reaches that, the messenger RNA is released, and then the amino acid chain that we just made will exit the ribosome, more than likely to go off to Gary Golgi apparatus so he can package and ship those proteins where they need to go. This has been Chapter 12, Part 5, on molecular genetics and DNA.